Before proceeding, please make sure to subscribe and turn on the bell icon for upcoming videos. Hello, and today's video is a continuation of the previous video, which was a detailed illustrative video on anatomy of the TMJ. You can watch it out for an even better understanding of today's video. That's going to be on histology of TMJ, including histology of articular surfaces, the intra-articular disc, and histology of the synovial membrane of the TMJ. Let's review some anatomic landmarks of the TMJ in this sagittal cross-sectional view. The structures shown in this lateral view are the glenoid fossa of the temporal bone, the condylar process of the mandibular bone, the intra-articular disc forming the upper and lower joint cavities that are filled with synovial fluid secreted by the synovial membrane. The synovial membrane occupies the joint spaces and lines the inner surface of the fibrous capsule and the margins of the intra-articular disc. But it does not cover the articular surfaces of the joint. Surrounding all these structures externally was the thin fibrous capsule of the TMJ, shown in green. Let's now move on with histology of these structures. The histology of TMJ in a child differs from that of an adult in so many aspects. That's why in today's video, the histology for both of them will be studied individually. The two unusual histologic features of the TMJ is that, unlike other joints, its articular surfaces, which are the glenoid fossa of the temporal bone and the condylar head of the mandible, are not covered by hyaline cartilage. Instead, they are covered by fibrous tissue. Second unusual feature is the presence of a secondary condylar cartilage located deep to the superficial fibrous head of the mandibular condyle until adolescence. And this is the main difference between the condyle of a child and that of an adult. Please watch my video on development of the mandible to have a more in-depth understanding of how and when this secondary condylar cartilage is formed. Let's now take a small section of the mandibular condyle first and study some histologic structures for both child and an adult. The most superficial layer of the condyle in both child and adult is formed by the layer of fibrous tissue. The main fibers in this fibrous tissue is collagen fibers, with a relative distribution of elastin fibers. This layer possesses sparse distribution of fibroblasts and fibrocytes. Beneath this fibrous layer is a more cellular layer called the cell-rich zone. In case of the condyle of a child, this layer is called the proliferative zone. The cells present in this zone function as progenitor cells, which possess undifferentiated and active chondroblast-like cells. These chondroblast-like cells elaborate proteoglycans and type II collagen to form the extracellular matrix of cartilage. The chondroblasts, after secreting the cartilage, becomes entrapped in the cartilage as chondrocytes, and this will be the third layer situated underneath the proliferative zone in the condyle of a child. This secondary condylar cartilage, just like cartilage elsewhere, will possess high amounts of type II collagen. In adults, what replaces this zone is the fibrocartilaginous layer. This fibrocartilaginous layer, as the name implies, will possess high amounts of collagen fibers. And a number of cells present in this layer will be round in shape, resembling cartilage-like cells. The most important part about this fibrocartilaginous layer is that although it's associated with the articular surfaces, it does not form part of the articulation and has got no formal functional role to play in the everyday movements occurring between the two bones of the joint. Immediately beneath this layer and covering the bone is a thin zone of calcified cartilage, both in condyle of an adult and a child. Unlike condyle of an adult, the condyle of the young child is not lined by a distinct layer of compact bone, and is due to the fact that the mandible of a child is still in its growth phase until adolescence, and absence of this compact bone allows free growth of the condyle. This histology of the articular surface of the mandibular condyle applies for the articular surface of the glenoid or mandibular fossa of the temporal bone as well, except for it being thicker at the articular eminence. Let's come to the histologic section of the intra-articular disc. The disc contains cells that are embedded in a matrix composed of collagen fibers and ground substance, and this is because of a decrease in the number of cells as the person ages. The intra-articular disc of the child possesses a huge number of cells compared to the sparse number of cells in the disc of an adult. The cells display a varied outline, varying between flattened or active fibroblast-like cells in young children and not so active and rounded or chondrocyte-like cells in adults. The ground substance of the disc comprises glycosaminoglycans, which accounts for about 5% of its dry weight. And the major components of glycosaminoglycans are chondroitin and dermatan sulfate. These components, due to their anionic charge, can absorb water and can help resist compressive loading. A unique feature of cells in this intra-articular disc is the numerous and fine processes of the cells, which extends for a considerable distance throughout the disc, both in an adult and a child. The processes are rich in gap junctions, allowing passage of nutrients and fluids from the periphery to its avascular central region. The majority of collagen fibers consists of type 1 collagen, with some traces of elastin fibers. Coming up next is the synovial membrane. The synovial membrane lines the margins of the intra-articular disc and inner surfaces of the joint capsule. It consists of two layers of cells, that is, a superficial layer of endothelial-like cells resting on the underlying vascular layer. The superficial layer consists of macrophage and fibroblast-like cells, the macrophage-like cells being phagocytic and fibroblast-like cells being productive in nature. I hope this video helps. Stay tuned for the next video, which will be about the anterior and posterior displacement of the intra-articular disc. If you think this video has really helped you, please do like and share the video. And don't forget to hit that subscribe button and turn on the bell icon to get notified on each upcoming video. Plus, if you have got any questions or suggestions regarding this video, you may write them down in the comment box. Thank you for watching.